This module will explore basic concepts related to neuron form and function, and we'll explore how these cells are especially adapted for very rapid signaling and communication within your bodies. Let's begin by taking a brief look at neuron structure. Now keep in mind that neurons come in a diverse array of shapes and sizes, but here we're taking a look at a basic neuron. Notice toward the far left of the cell we have dendrites, which often receive signals from other neurons or various parts of your body. We have a cell body. This is the part of a neuron that actually contains the nucleus of the cell. We then have this nice long axon here, which when stimulated can send an electrical impulse very rapidly down along the length of this structure. When that impulse reaches the end of the axon, it reaches these terminal branches toward the far right. Those terminal branches can then communicate with other neurons or with muscles, and often causing muscle to contract by releasing neurotransmitters. So these synapses are essentially junctions between the terminal branches of one neuron and an adjacent neuron or a muscle cell. So let's explore some basic concepts related to how these neurons actually function. So how do neurons work? Well, some basic key concepts to keep in mind are as follows. Neurons at rest, in other words, a neuron that is not firing or not sending a signal, tends to be polarized, meaning the inside of the cell tends to have a negative charge relative to its external surrounding environment. So again, we say neurons at rest are polarized. Also, we have sodium-potassium pumps at work. So sodium-potassium pumps, these are proteins embedded in the membrane of neurons. They are undergoing active transport, so they require ATP. They are actively pumping sodium ions out of the neuron and potassium ions in the neuron. So again, this is all taking place when neurons are at rest. When we're talking about the signal that neurons can send, it's in the form of an electrical impulse. So when a neuron is stimulated, we could say it fires or it sends a signal. That tends to be a depolarization that rapidly propagates itself down along the axon, and we refer to this as an action potential. So again, neurons communicate by sending these action potentials down along the length of their cells. Also, keep in mind, we'll illustrate this in just a second, but keep in mind that there are other proteins, we call them voltage-gated channels, that allow the movement of specific ions, for example, either sodium or potassium, across the neuron membrane. But these proteins, these channels, tend to open only in response to a depolarization. When a cell is polarized, in other words, when a neuron is at rest, these proteins tend to be closed, and therefore they don't typically allow the movement of the sodium or potassium ions. So let's illustrate these concepts in another fashion. So what I've done here is just simply sketched out a portion of an axon, which of course is part of a much larger neuron. So just to illustrate some of these key concepts. Again, we're looking at a small portion of an axon. Keep in mind that overall the cell itself tends to have a negative charge relative to its surrounding environment. And again, that's due in part to this sodium-potassium pump illustrated right here. So again, this is a protein undergoing active transport, pumping sodium ions out of the neuron, potassium ions into the neuron. It tends to pump more sodium than potassium, and there are other ions at work as well. So again, overall, the cell tends to have a negative charge compared to its external environment. Here are just a few of these voltage-gated channels that we mentioned. So these, again, are proteins embedded in the cell membrane. As they're sketched right now, they're closed. So ions cannot move across membrane, but in response to a depolarization or an action potential, these proteins would tend to open and again allow either the rapid diffusion of sodium into a cell or potassium out of the cell. So essentially what we have here is a situation in a neuron at rest. Sodium pumped out, potassium pumped in, and overall the cell has a negative charge or a resting potential, again, compared to its external environment. So we mentioned previously that a neuron at rest is polarized. So it might have a resting potential of negative 65, negative 70 millivolts, somewhere in that region. Then what type of signal travels along a neuron when it is stimulated? Well, again, it's in the form of this action potential, which again is simply a rapid depolarization that propagates itself down along the length of the axon. So let's take a look at the events that take place during the generation and propagation of an action potential. What we see here is essentially the electrical activity or the electrical membrane potential of a neuron as it propagates an action potential. So let's explore what is happening. So imagine our cell at rest. If stimulated, one of the first things that takes place is that those voltage gated channels, specifically the sodium gates, they open. In response to the opening of those sodium gates, sodium rapidly diffuses into the cell. When that occurs, the membrane potential becomes depolarized or becomes rapidly moving in the positive direction. So again, this first phase is this rapid depolarization due in response to the sodium gates open. Split second later, those potassium gates open. Potassium gates open, that allows the potassium ions to flow out of the cells. That repolarizes the neuron. Those gates can remain open just for a split second longer than they should perhaps. Often we see this undershoot where the cell actually becomes more negative than it was to begin with. But then very rapidly the sodium-potassium pump again is doing its thing. It's actively transporting sodium out, potassium in, thereby reestablishing the resting potential of that neuron. So just a few things to keep in mind about action potentials. First of all, these are all or nothing events. A cell either sends an action potential or it does not. It does not occur in a graded fashion. Also, keep in mind that in order for a cell to generate and propagate an action potential, the cell must be depolarized to some threshold level. So for example, in this image here, let's say the resting potential is negative 70 millivolts. The threshold is maybe negative 55 millivolts. So and again, in order for this cell to propagate an action potential, it must be depolarized to negative 55 millivolts or above, thereby generating an action potential. So once that action potential reaches the end of a neuron, it reaches the terminal branches, how then does that neuron communicate with other cells? Well, now we can discuss synapses and the role of neurotransmitters. So imagine we have two cells communicating. Here we might talk about a presynaptic neuron generating an action potential, sending it down to the terminal branches where it communicates with a postsynaptic neuron. What occurs in that synaptic region? So imagine an action potential making its way to the end of a terminal branch. When it reaches the end of the terminal branch, that triggers these vesicles to release neurotransmitters, illustrated by these small blue circles. Neurotransmitters then can bind with receptors, these green structures, located in a postsynaptic neuron. When they bind with those receptors, that can cause those receptors to actually open. So much like the voltage-gated channels we mentioned previously, here we're talking about receptors that can open and allow the diffusion of specific ions. So for example, if a neurotransmitter is released and it happens to bind with receptors that allow the passage of sodium ions across the cell membrane, what would happen? Well, sodium would rapidly diffuse into this postsynaptic cell, thereby tending to cause that postsynaptic cell to depolarize or potentially generate an action potential. On the other hand, what if neurotransmitters are released that bind to receptors that, when open, allow the specific diffusion of potassium? In that case, potassium would diffuse out of this postsynaptic cell, which would tend to lower the membrane potential of the postsynaptic cell, which would essentially tend to prevent that postsynaptic cell from generating an action potential. So we can talk about these in terms of excitatory responses or inhibitory responses. So neurotransmitters, again, can cause excitatory postsynaptic potentials, or EPSPs. This essentially is the case when a neurotransmitter tends to depolarize a postsynaptic cell, as we see toward the left, or neurotransmitters can cause inhibitory postsynaptic potentials. And again, this is a situation where a neurotransmitter tends to polarize a postsynaptic cell even further, or bring its membrane potential even more negative than you began with. So, in essence, we can have these two different types of signals being sent to postsynaptic cells. What happens then if a postsynaptic neuron receives signals from multiple neurons at the same time, some of which might be excitatory, while others might be inhibitory? So that's illustrated toward this lower left image. So a single large postsynaptic cell here, again, receiving multiple signals at the same time. In essence, the basic process we call summation. So if multiple signals arrive at the same time, all you have to do is essentially add up all of the excitatory signals, subtract all of the inhibitory signals, and if overall that tends to depolarize this postsynaptic cell past its threshold level, then yes, sure enough, that postsynaptic cell will generate an action potential. If not, then the cell will not fire. Again, this basic process is simply referred to as summation. That concludes our very brief discussion of